All right, I'm going to turn up the microphone. Can you guys hear me okay? Sorry, my voice is all jacked up. Some stupid virus. But hate that. All right, so today we're going to talk about replication. Remember we were doing DNA replication, whatever day that was, Thursday? DNA replication physiologically is for when cells need to reproduce themselves. Right? If you need to make 2 more cells because you got a cut and your skin needs to heal over, or you need more liver cells or brain cells, which would be nice for us, or if your immune system needs to sort of, make some more white blood cells to fight whatever virus happens to be taking over your mucous membranes, then you need to first do DNA replication so that the 2 cells that come from the original have the same amount of DNA, right? If a cell starts with 46 chromosomes and then it splits in half to make 2 cells, you want each of those cells to have 46 chromosomes, right? Every single cell in our body needs to have all the genes, all 46 chromosomes, 2 copies of every gene in all of our cells. The only exception to that is in eggs and sperm. Okay? Those are the only 2 cell types that have half the amount of DNA. Everything else needs all 46. So what evil scientists have done is taken the process of replication and repeated it in a test tube. Okay? The polymerase chain reaction is just DNA replication, but done in a test tube instead of in your cells. Okay? So we figured out all the enzymes that are needed to do DNA replication, all the other stuff, right? All the nucleotides, all of that crap, and we can do that now in a test tube to amplify or make more DNA. Okay? So the process of PCR, which is the polymerase chain reaction, which we're going to talk about, has revolutionized our ability first to just study genes and so forth, in science for health and all that, but also for personal identification of individuals. Okay? So we're going to talk about PCR and then how it's used to solve crimes and to figure out who's the daddy and all that kind of stuff too. Okay, so we talked about in the video of replication from last week Thursday, the real one, the educational one, I said you guys don't need to know all the details of replication, right? You need to know that it occurs by complementary base pairing, right, on both strands of DNA, right? Every single chromosome is really double-stranded DNA, a big, huge, long piece that then's all wrapped up on itself to make that little sausage guy, right? The 2 strands stick together because of complementary base pairing, which is A's and T's stick together, G's and C's stick together, T's and A's, C's and G's, right? All the way across. We separate that, and then that enzyme DNA polymerase adds nucleotides by complementary base pairing. If it sees an A, it adds a T. If it sees a C, it adds a G. If it sees a T, it adds an A. If it sees a C, it adds a G, and so forth, all the way down to make an exact copy. Now, in that little video, they talked about you need a primer to do that. The little piece of RNA has to stick in there to get it going, and all this other baloney that had to happen, which you guys don't need to know for regular DNA replication. Okay? But in order for scientists to come up with a way of doing this in the test tube, they did have to understand exactly how it works. And what does that DNA polymerase enzyme need, right, to actually do replication? And so in order to do replication in a test tube, right, you need to have those free nucleotides, right, the A's, the G's, the C's, and the T's, those molecules, in order to add to do complementary base pairing, right? Those have to be available, just like in regular replication. Right? We need this same DNA polymerase enzyme, right? Because that's the thing that does that actually adds the base pairs to build that DNA molecule. And then we have to have primers to get it started. And those are, in this case, are just little pieces of already made DNA that's complementary to your template. Okay? So for PCR, we don't have to make the entire chromosome. Whereas in DNA replication in a cell, right, they're replicating the entire chromosome of all of our chromosomes. If we use PCR, we can replicate just a small part of a chromosome just by where we place these little primers, right? We know the entire sequence of the human genome, right? We talked about that last time. We've sequenced the whole genome. We know what every single base pair in order on all 46 chromosomes. So any genes that have already been identified, like insulin, for example, we could just design these little primers to be complementary to either side of the insulin gene, one going that way, one going that way, and we can do DNA replication of just the insulin gene, okay, using this PCR. So we can make a whole bunch of that insulin gene DNA, and then we can study it or use it to do other stuff. Okay. So for PCR, we don't have to make the entire chromosome. For regular DNA replication that happens in your cells, we do want to make the entire chromosome. But here we can just make individual genes or just small parts of the chromosomes, just by how we make these complementary to that little sequence. So we can sort of target what genes we want to do with PCR. Okay? But we still need all the stuff excuse me, that the cell would need to do replication in order to do this process. Okay. Okay. And so here's our little test tube that we do all this in. First, we need DNA from we need the template DNA, right? So we need something that we're going to do this PCR off of, right? So if I wanted to PCR out one of your genes, right, I would need to take some cell samples from your cheek, right? If you just swab the inside of somebody's cheek with a Q-tip, you can get enough cells on there to extract the DNA, and then we could stick it in a test tube to do PCR. Okay? And then we add our nucleotides. Remember, these are the bases, the nucleotides A, T, C's, and G's, those molecules, those are the ones that we stick in there to build up. DNA polymerase enzyme, right, which has been purified. In this case, we use enzyme from bacteria because it's easier to get from them. And if we go back to all the crap we learned about evolution, we all use the same DNA as our genetic material, bacteria and us, right? The exact same nucleotides. Almost the exact same enzyme. For our purposes, DNA polymerase in a bacteria works exactly like the DNA polymerase in your cells. So we can get it from a bacteria, right? And we can get it from a bacteria that lives someplace like in the ocean in those thermal vents where the water's boiling. And so that enzyme is very stable even at high temperatures. Most enzymes totally suck at high temperatures or low temperatures. They die, they fall apart, they don't work. So ours, if we extracted it from your cells and had a bunch of this enzyme and tried to manipulate it in the lab and students carrying it around and it's not on ice or it's not at 98.6, it wouldn't work anymore. But we can get it from a bacteria because we all use this same system to do replication. So we use a bacterial excuse me, polymerase, and then we have to add these little primers, little pieces of DNA, AGCT, about 20 base pairs, little tiny piece that's complementary to the region wherever we're trying to amplify. Okay? Instead of doing it from the ends of chromosomes and amplifying a billion base pairs, we can just do PCR on 300 or 1,000, just a small piece, right? Just the insulin gene, or whatever we're looking at. And so then here's our little picture. Again, I want you to understand conceptually how PCR works without me necessarily asking you specifics. You should know that it needs the DNA polymerase, it needs the primers, it needs the nucleotides, and you have to have a template. The same exact stuff that we need for regular replication. Okay? So we can make billions of copies of this little piece of DNA from a very small amount of starting material, right? If we scrape just a little bit of cells from your cheeks, we can get a tiny bit of DNA. You could never see it, right? We do a DNA extraction on those cells, and at the end it would look like, a couple drops of water. And then, okay, well, hopefully it's in there. And then from that, teeny tiny little few drops of water, you can make enough DNA so that you could actually see it look like a white little stringy thing in a test tube. Okay? If you take enough cells, like if I took an entire strawberry and mushed it up and extracted DNA, we could see it. It looks like spider webs because there's so much in there. But that's because the strawberry is made out of 1 billion cells, right? If you're only taking 30 cells from your cheek, you're never going to see it. So a teeny tiny amount that we can amplify, because all we really need is one molecule of DNA to get our primers to sit on. DNA polymerase will make a copy of that, and then we do that cycle again. We do replication again, and then we make copies of that, and over and over and over and over. Okay? And so then every time you make a new little piece, the primers can sit on that and amplify the original template and the newly made one. And so if you run it through 30 cycles, you can make billions of molecules of DNA from a very, very little bit that you'd start with. Excuse me. And so to do this, they just heat it up, they cool it down, they heat it up, they put it at the different temperatures that the enzyme works, right? We get the primers to sit, the enzyme sits on there, it does complementary base pairing, we heat it up, everything falls apart, right? Just like in replication when the two strands come apart, right? Then we can have the primer sit, do DNA polymerase, complementary base pairing, heat it back up, fall apart, and over and over and over and over, so you can make tons and tons of a little piece of DNA.